When talking about the antigen-antibody interaction, we must first know about the combining site of antibody, which is a cleft and roughly 3×1 nanometer. That is the size of 5 to 6 sugar units. Although there is evidence that antigen may bind to larger or even separate parts of variable region. The binding depends on a close three-dimensional fit, allowing weak intermolecular forces to overcome the natural repulsion between molecules. Although binding between antigen and antibody involves only non-covalent forces that are very strong and irreversible. However, in practice, the high affinity of most antibodies means that they become rarely detached from the molecule when they are being destroyed by phagocytosis. So let us discuss some aspects of antigen-antibody interactions. We have van der Waals forces, hydrogen bonding, electrostatic interaction, hydrophobic region interaction, affinity profile, and antigen binding. If we talk about van der Waals forces, then they attract all molecules through their electronic cloud, but only act at extremely close range, which means there are weaker forces. When we talk about the hydrogen bonding, then it is also a kind of a very weak force, and it is only present in amine and hydroxyl groups. When talking about the electrostatic interactions, they are between parts of an antibody and antigen molecule, with a net opposite charge on both of them. The positively charged is ammonium group and the negatively charged is carboxyl group, and they are the strongest forces between them. Talking about the hydrophobic region interactions, then they are the most strongest ones. It basically attracts the aqueous environment and which is going to produce a strongest forces of interaction. Well, next we must discuss about the affinity profile, and it is normally expressed as high affinity or low affinity. Affinity is normally expressed as the association constant under equilibrium conditions. A value of 105 liter per mole is considered as low, while the value of 1010 liter per mole is considered as a high affinity. High affinity antibodies are affected in most cases, but low affinity antibodies persists too, and may have certain advantages, for example, reusability and high tolerance, while higher affinity do not have those. Well, in practice, the avidity is most oftenly measured because antibodies have at least two valencies. And even with monovalent antigens, a serum can only be assigned an average affinity. The average affinity tends to increase with time after antigenic stimulation, partly through cell selection by diminishing amounts of antigen. Now let us talk about the antigen binding. Well, it is remarkable that the process of making antibodies is so versatile that one can find antibodies specific for almost any type of molecular surface. These include the most common component, such as proteins, nucleic acids, sugar, and lipids, but also man-made synthetic molecules as well. For example, antibodies against amphetamines are being developed for the treatment of drug addiction. Well, that was it for the antigen-antibody interactions. Stay with us and learn about immune complexes in a bit.